I know there's a lot on this slide, but I promise you stick with me, and by the end you will know exactly how fatty acid synthase works to synthesize saturated C16 palmitic acid in mammalian cells. Okay, so just as a reminder, another video that sort of walks through the details for kind of the chemistry that happens. So looking at the chemistry of loading our electrophiles and nucleophiles on fatty acid synthase, sort of doing chemistry in the Ks domain, and then dipping into our other domains on fatty acid synthase to do our reduction, dehydration, reduction, finally cleaving off our fatty acid from our finished fatty acid from fatty acid synthase, and then doing any modifications as necessary to make them longer, introduce units of unsaturation, so forth. What we're focusing on in this video here is basically just the chemistry that happens sort of here, understanding how fatty acid synthase does this. So a couple pieces with this, and then after I sort of walk through these parts, we'll have sort of a video that we'll watch that I'll sort of help narrate through that sort of puts a lot of these pieces together. But here is the structure of fatty acid synthase. So it is a multi-enzyme complex. So much like we see for pyruvate dehydrogenase, here's how it's different. Pyruvate dehydrogenase is multiple enzymes that are separate, subunits that come together. This is really all of these kind of in a strip mall sense, all strung together. You'll notice there's this gray section in the middle that says dimerization. Fatty acid synthase exists as a dimer. So we're going to have two of these sort of ribbon strips come together, and then what we're going to see, it's going to fold into a structure, we'll get to that when we watch our video, that basically puts these domains into different regions. And I want you to think of it like a basement level that we're going to be doing our condensation chemistry, and then an upper level that we're going to be talking about doing all of the rest of the functional group modifications that we need. Okay, so this guy is fatty acid synthase. Okay, so that enzyme is fatty acid synthase. We're going to see it as a dimer, so two of these are going to come together. We're not going to talk about rate controlling steps of fatty acid synthase, because the rate controlling step for fatty acid synthesis, remember, is acetyl-CoA carboxylase, ACC chemistry. That is what it's all about. So we haven't yet kind of brought that piece into this picture, but acetyl-CoA carboxylase is going to be necessary for synthesizing malonyl-CoA. Okay, so there's another video on sort of ACC chemistry, but that chemistry is really important because it's going to make malonyl-CoA. We need to have malonyl-CoA to couple with acetyl-CoA to do this chemistry. Okay, so a couple of pieces I'm going to zoom in here that I want to highlight that we're going to sort of refer to periodically as we talk about this. We're going to see this acyl carrier protein and then this long arm, this pantothene arm. Okay, so this is going to be what I'm referring to as the long arm. We saw that in pyruvate dehydrogenase chemistry. We had that long arm that allowed us to dip back and forth between different domains. We're going to see the same thing here. I also want to point out there's this Ks domain. Okay, so this is another domain. These are a couple of the domains that I do need you to know by name. Okay, so this Ks domain, you'll notice that it has a cysteine, so it has a thiol group there. Okay, this is going to become important on the bottom of the slide here. I'm going to highlight in a second where we see all our sulfur-containing sort of functional groups, and so how we are going to utilize those. But when we start doing our chemistry, we're always going to have our electrophile is going to be sitting in this Ks domain, okay? And our nucleophile, when we start doing our condensation chemistry, is going to be in this acyl carrier domain with this long arm. Okay, so when we start doing chemistry, we're going to have our acetyl-CoA group over here on the Ks domain as our electrophile, and we're going to have our malonyl-CoA over here on this long arm. Okay, this will make a lot more sense once we go for a video where you can sort of see all of these pieces coming together. Okay, but what I'm going to do is then go through these different steps that happen: priming, condensation, and then we see the reduction, dehydration, reduction, cleavage chemistry that we saw on our previous video. So before I do that, I just want to make sure to remind everybody places where we see thiol groups. Okay, so I'm going to just kind of put this down here as a way. Don't have a lot of space on this slide, so where we're going to sort of put this in here. So this is going to be where we see our thiol groups. Okay, so here's where we're going to have our thiols. Okay, 3 places that we're going to see thiols here. Okay, we're going to see CoA has a thiol group. Okay, acyl carrier protein has a thiol group, and then our Ks protein is going to have, via a cysteine residue, a thiol group. Let me just zip back here just for a quick second in case you hadn't watched this video yet. When we talked about our tagging and activation step, here you can again see the structures. So this is not a thiol group that we have as part of a specific cysteine residue in a protein, but acyl carrier protein and CoA sort of have these cysteamine sort of and ending groups, and that's how we have that thiol. Okay, so if this is not familiar, make sure to go back and watch that tagging and activation video. Okay, so here we go. That's where we're going to see our thiol groups. Okay, and over here I just want to remind us of who is going to be our electrophile and who is going to be our nucleophile. Okay, so we are going to have, again, a thiol containing group. We're going to have this acetyl group on acetyl-CoA. Okay, and that is going to be our electrophile. So here we've got acetyl-CoA. So we've got a C2 group that we're going to have. And then our nucleophile is going to be malonyl-CoA. So again, sulfur there. And then what we're going to have is this guy. And I'm going to put this carboxylate group in a different color here, because this is what's going to come off as CO2 when we generate malonyl-CoA. Okay, so what's going to happen, I'm just going to highlight this here. We are going to have the loss of CO2, and that's ultimately going to put a pair of electrons here and make this guy be our nucleophile. So that's how that chemistry works. Remember, malonyl-CoA is a beta keto acid, and so we're going to be able to use it in that sense to decarboxylate and generate the nucleophile that we need. Okay, so let's talk about excuse me, all the different pieces that we have here, the chemistry that's happening. Okay, so we basically have 2 substrates that we need to prime and load, and I think I referred to this on the previous slide as loading. So I'm just going to put this here because we sort of talked about that as being sort of the loading step. We're loading our building blocks onto fatty acid synthase, and so we're going to have acetyl-CoA and malonyl-CoA. Okay, acetyl-CoA being that C2 piece, malonyl-CoA being that C3 piece. Okay, nothing's really necessary to kind of load these guys onto things. The synthase domain that we're going to see. Now, I should say that the first thing that happens is everybody starts out on the acyl carrier protein. Okay, so this is going to both be acetyl-CoA and then malonyl-CoA. Okay, but what's going to happen then is we are going to transfer the acetyl-CoA to the Ks domain. So even though, and you'll see this when we watch the video, we're going to start out, everybody starts out by being put on the acyl carrier protein, and then we're going to put you on the Ks domain. Okay, when you're ready to be doing condensation, you're going to be on that Ks domain because that's where chemistry happens. So again, the purpose of what's happening here, we are basically loading, there's that loading word, loading our building blocks. Okay, all right, next step that we have here, this is where we do our chemistry step, our condensation step. Okay, so what we're going to have is we're going to have our electrophile. That's going to be our acetyl group. So our acetyl group Okay, that's going to be our C2 piece. And then we're going to have our nucleophile, which is our malonyl group. That's our C3 group. Okay, no cofactors are necessary specifically here, but really what we see is we see a negative CO2. We're going to lose CO2, and that's what allows us to make malonyl-CoA, essentially a 2-carbon nucleophile. Where this is happening is our Ks domain, okay? And what happens is we're going to have the acyl carrier protein reach over to kind of put those together. So look back to our priming step, right? Our loading step. We basically had our nucleophile and our electrophile on 2 different domains. So if those guys need to come together, what has to happen is that acyl carrier protein that has the malonyl-CoA has to dip over to the Ks domain, and our chemistry is going to happen. So purpose, again, of what's happening here is we have our carbon-carbon bond form that happens. So carbon-carbon bond formation. Okay, and then we've added a C2 unit. So we've coupled 2 C2 units, or as we're going to see as we repeat this, we basically continue to add 2 carbon units. Okay, so that happens in what we're going to refer to kind of as the basement part of our fatty acid synthase. And then what's going to happen is we're going to have our acyl carrier protein dip up into the top floor where we're going to be doing our reduction, dehydration, reduction. Okay, and then we're going to keep repeating that until we're done. So I'm not going to say that there's any specific substrates here for what we have for this reduction. Here's some cofactors that we need. To do that first reduction, we need NADPH. You do not need to know these names. I'm going to write them in here, but I'm going to highlight in just a second. These are not names that you need to know. So what's happening is we're basically taking that carbonyl and we're converting it into an alcohol. So that's the functional group changes that are happening when we do this first reduction step. Zoom out here so we can make sure to see all of this. So second step is we're going to see the loss of water. So it's not really a substrate that's needed or a cofactor that's needed, but it's something that's eliminated. Okay, I'm going to write this down again. You do not need to know. Let's put this in here so we understand. All of these guys you do not need to know. Okay, you do not need to know names. Do not need to know names. Okay, chemistry that happens here is we're going to have an alcohol that's dehydrated to make an alkene. We have another reduction step. Remember, both of these reduction steps happen with NADPH. Just writing this in, you do not need to know the enzyme names. That takes our alkene to an alkane. And then what's going to happen here is if this is the first time we've gone through this, we've taken 2 plus 2, we've made 4. We need to repeat this, okay? So what's going to happen is we're going to go back up here and we are going to repeat. Okay? So this is going to be repeated 7 more times, and that ultimately is going to get us to a C16 fatty acid. Okay, so we'll see this when we look at the video too, but what's happening is we've got our acyl carrier protein that dipped in and coupled to that 2-carbon piece. Our acyl carrier protein now has a 4-carbon piece, but not with the right functional groups. So we're going to dip up into the upper floors and do those reductions, dehydration reductions, and now we've got a 4-carbon piece that looks like a little 4-carbon saturated fatty acid. So we'll see these details when we look at the enzyme active site, but we're going to repeat this process, but now we're going to be adding 2 more units. Okay, we're going to keep this happening until we get to our C16 fatty acid, and then that C16 fatty acid is subject to sort of this cleavage chemistry. This is an esterase that's actually going to be doing chemistry, so it's going to need to have water to hydrolyze that ester linkage. And basically what's going to happen in this TE domain is we are going to release that C16 fatty acid from fatty acid synthase. Okay, So I know that that was a lot there, but what we're going to do now is we're going to transition into me narrating over another video. So it's going to be something that we'll play, we'll pause, we'll kind of talk about how it reflects and refers back to this chemistry that we've talked about here. All right, I'm going to walk through, and you can watch this video. It's only a 5-minute video here. It might take me 10 minutes to go through it because I'm going to play and pause so that we can sort of revisit things that we've talked about and kind of make sure to reinforce them as this video plays. But I encourage you to go back, find this on Blackboard, and watch it yourself.
Glycogen and triglycerides are the two main forms of energy storage in the body.
So, again, I didn't get too far and I'm already pausing it. Remember, we've talked about the main purpose of carbohydrates and lipids are to be fuels. So they're going to talk about proteins here in a minute. But glycogen, right, is our storage polymer of glucose, but we have limited reserves of that, 12 to 24 hours probably at most. And, once we run out of that, we don't have any more glucose that we can make other than by gluconeogenesis. Triglycerides, relatively unlimited storage. So kind of being able to take excess fuel and store it as triglycerides in adipose tissue puts it in the most sort of cost-effective space. But then being able to tap into that is going to be really key. And we're going to see how insulin is really important in that process.
Triglycerides are hydrophobic molecules composed of a glycerol backbone and 3 fatty acids. In order to synthesize triglycerides, the body must first make fatty acids. In this video, we will talk about fatty acid synthesis, which is catalyzed by 2 enzymes, acetyl-CoA carboxylase and fatty acid synthase.
So pausing again for a second, acetyl-CoA carboxylase, which we're going to call ACC, this is the rate controlling kind of step in this process. So ACC is responsible for making malonyl-CoA, okay, and we're going to see how it does that. I'll pause because we've learned a lot of details for how that happens. And then here is fatty acid synthase. This is our and I'm not going to say it's not really a multi-enzyme complex because it's really just one enzyme as a dimer that has multiple domains. So it's different from, say, pyruvate dehydrogenase that we've learned about before that really has actually multiple different proteins. This is kind of like this, they're all strung together here. Okay.
Following ingestion of excess carbohydrates or proteins.
This is really important here. We've highlighted that this is not from excess fat consumption. You're not going to take excess fat, break it down into acetyl-CoA, and then remake it into fatty acids. You just store it directly as that fat. But highlighting that proteins too are not meant to necessarily be metabolized as energy. They're building blocks, but if you have excess, it does need to be metabolized.
Fatty acid synthesis takes place in the liver. Carbohydrates and proteins both provide carbon skeletons in the form of glucose and amino acids, which, in the presence of insulin, can be oxidized to acetyl-CoA, the starting material.
Okay, a lot that they went through there. I'm going to probably rewind it and play it too. So again, carbohydrates are broken down into glucose. Amino acids, right, can be broken down into their sort of carbon skeletons that we're able to metabolize. You'll see that things went into the mitochondrion here. Pyruvate dehydrogenase, and again, in this case, we're talking about the endpoint of glycolysis as pyruvate is going to generate acetyl-CoA. Okay, you'll notice here that this only happens kind of in the presence of insulin when we are in a fed state. That ensures that you don't have glucose that was derived from glycogen breakdown or gluconeogenesis kind of going into this process. It would be sort of a futile cycle. So let me just rewind this a little bit and I'll not talk and I'll let it sort of play out here.
Of excess carbohydrates or proteins, fatty acid synthesis takes place in the liver. Carbohydrates and proteins both provide carbon skeletons in the form of glucose and amino acids, which, in the presence of insulin, can be oxidized to acetyl-CoA, the starting material for fatty acid synthesis.
Okay, so they're going to talk about here, we've mentioned this, there is not a way, so this breakdown, whether it's pyruvate dehydrogenase chemistry or if we were talking about beta oxidation, right? All of these breakdown processes happen in the mitochondria. Once we have acetyl-CoA in the mitochondria, we need to think about how we are going to be getting it out into the cytosol, because that is where fatty acid synthesis occurs. So they're going to highlight here that we do not have transporters for acetyl-CoA. We need to disguise it as something. What we're going to see is they're going to disguise it as citrate. So you're going to talk about dipping into citric acid cycle chemistry using citrate synthase to synthesize citrate. This is going to be that citrate transporter. We're going to see the citrate come out here, and then we're going to regenerate our acetyl-CoA using an enzyme called citrate lyase.
Acetyl-CoA is made in the mitochondria, but is unable to cross the mitochondrial membranes to the cytoplasm where fatty acid synthesis occurs. To get around this, citrate synthase combines acetyl-CoA with oxaloacetate to form citrate. A citrate transporter brings the citrate into the cytoplasm where ATP citrate lyase splits citrate back into acetyl-CoA and oxaloacetate.
Now, one of the things they don't mention here is we can get that oxaloacetate, because we're in a fed state. We're not using that oxaloacetate for gluconeogenesis. We can use the malate aspartate shuttle to get that oxaloacetate back into the mitochondria so it can continue to do this citrate export. Okay, so I'm going to highlight here. So here's our acetyl-CoA. So noting some important pieces of this, we've got 2 carbon piece as an acyl group on CoA. We've got that important thioester bond.
Each molecule of acetyl-CoA contains 2 carbons. In order to create a long-chain 16-carbon saturated fatty acid, 2 carbons are added at a time to build the chain. Acetyl-CoA carboxylase carries out the activation step.
So remember, we've talked about sort of this activation step. This is not able to be a nucleophile. If we're going to be doing carbon-carbon bond-forming chemistry, somebody needs to be a nucleophile, somebody needs to be an electrophile. The net is we're adding 2 acetyl-CoAs together, but one of them needs to be a nucleophile, one of them needs to be an electrophile. What we're going to be doing with acetyl-CoA carboxylase, which we're just going to call ACC, this enzyme is responsible for converting acetyl-CoA into malonyl-CoA. What we're going to see here is it's going to add another carbon atom on here. It's going to add a carboxylate group on the end. That's going to generate the beta-keto acid malonyl-CoA. And why that's important is when we do our chemistry step, when we're doing our condensation step in fatty acid synthase, we're going to let that CO2 come off, and it's going to generate a nucleophilic carbon atom right here so that we can do our carbon-carbon coupling step. But they're going to show the chemistry here that happens with ACC. What we're going to see is a vitamin B7 that we're going to have here with biotin. We're going to see that as a way to create that transferable CO2.
An irreversible, committed step in fatty acid synthesis. In this step, acetyl-CoA is activated using ATP and bicarbonate, which converts it to the 3-carbon malonyl-CoA.
So we saw a lot that happened there, right? We saw bicarbonate come in. That was our 1-carbon source. We needed ATP. This is not a chemistry that happens without the input of energy. They didn't highlight, but it's vitamin B7 as biotin that's responsible for doing this carboxylation step, and we generate malonyl-CoA. So here again is our beta-keto acid. Okay, so in malonyl-CoA here, what's going to happen when malonyl-CoA wants to be a nucleophile? We can decarboxylate it, and then these electrons are going to reside on this carbon atom here, and that's going to allow that carbon atom to be nucleophilic.
Malonyl-CoA is the activated carbon donor used by fatty acid synthase to elongate the chain. Fatty acid synthase is a multi-enzyme complex that extends the carbon chain to a 16-carbon saturated fatty acid. Mammalian fatty acid synthase functions as a dimer, where each identical subunit contains 7 catalytic domains. Fatty acid synthase has 2 major compartments. Transfer and condensation reactions occur in the lower compartment, while modification of the growing chain occurs in the upper compartment.
So again, this looks like a whole giant bananas piece, right? But I want you to think about there being two domains. This is what we were calling sort of the basement domain. That's sort of where we do our condensation chemistry, and then we do our modification sort of up here. He's going to talk in just a second about these two domains down here. This little guy here is going to be our long arm. That's going to be our acyl carrier protein. And then this domain here is our TE domain, which again, you didn't need to know the name of that, but that's going to be how we snip off our fatty acid when it's finished doing all of its growing that we have here. Okay, so this is a dimer, so there's like two pieces that sort of come together here with some pretty beautiful symmetry. This is what happens with mammalian fatty acid synthase. He's not going to talk about it here, but if you talk about fatty acid synthesis that occurs, say, in prokaryotic bacteria, these domains actually are individual enzymes, which allows for unique ways to sort of regulate some things or kind of influence some things with bacteria.
In addition, two other proteins, acyl carrier protein and thioesterase, are associated with the complex. To begin synthesis of the fatty acid chain, a priming reaction involving the enzyme malonyl acetyltransferase attaches the first acetyl-CoA to the acyl carrier protein and releases CoA. Acyl carrier protein contains a prosthetic group called phosphopentathene that creates a covalent thioester bond with acetyl-CoA. Next, the two carbons are transferred to the condensing enzyme beta-ketoacyl synthase in order to free up the acyl carrier protein.
Okay, so there was a lot there. I'm probably going to rewind this after I speak to it, and we'll just kind of show this again. Acyl carrier protein is going to be this long arm that sort of reaches back and forth. We need to load things onto it. And we didn't learn about the enzyme name here. You don't need to know about this. But this enzyme, this MAT enzyme, is what's responsible for loading things on acyl carrier protein. When we have something that we want to do some chemistry with, when we have something that we want to essentially be our electrophile, that's going to be over here in our KS domain. Nucleophiles are going to be on our acyl carrier protein. And then once we do that condensation chemistry, this acyl carrier protein will take that to the upstairs domain where it's going to have some other things happen. So let me rewind. Now that I've said that, we'll kind of show this piece again.
Synthesis of the fatty acid chain, a priming reaction involving the enzyme malonyl acetyltransferase attaches the first acetyl-CoA to the acyl carrier protein and releases CoA. Acyl carrier protein contains a prosthetic group called phosphopentathene that creates a covalent thioester bond with acetyl-CoA. Next, the two carbons are transferred to the condensing enzyme beta-ketoacyl synthase in order to free up the acyl carrier protein. Malonyl acetyltransferase attaches the first malonyl-CoA to the acyl carrier protein and releases CoA. The two carbons are...
So again, now we've loaded malonyl-CoA on here. So that's our priming reaction. And now we've got our nucleophile here and our electrophile ready to do chemistry. So that's what they're going to talk about sort of in this next piece here.
Malonyl acetyltransferase attaches the first malonyl-CoA to the acyl carrier protein and releases CoA. The two carbons, originally from acetyl-CoA, which are now on beta-ketoacyl synthase, are then transferred to the...
So I'm going to just stop here for a second because they show this chemistry, which is right, but they don't really talk specifically about it. So here is our malonyl-CoA. It's going to decarboxylate, which is going to generate a nucleophile on this green carbon here. Now, that's going to nucleophilically attack this carbon atom. And again, I'm drawing mechanistically here. We're going to have our electrons come up, come down, and then that's going to release essentially this thiol group on this KS domain, right? That's actually a cysteine residue on that protein. That's going to eliminate essentially what equates to a good leaving group there. So I'm going to rewind this just a second so we can see this. So we're going to see carbon dioxide be eliminated. That makes this green carbon, which is the CH2 group here, be nucleophilic. Nucleophilically attacks this carbon atom of essentially this acetyl group that was on KS, and we're going to couple those together.
CoA, which are now on beta-ketoacyl synthase, are then transferred to the end of the two carbons donated by malonyl-CoA in a condensation reaction. This condensation is made favorable by the decarboxylation of malonyl-CoA. The 4-carbon growing chain is now covalently bound to the acyl carrier protein and is ready for chemical modification.
Okay, so we finished the basement chemistry. The basement chemistry is that condensation. That's where we're combining nucleophile and electrophile to make a carbon-carbon bond. Okay, now we've got chemistry here that is not what we need it to be. So in these red sort of carbon atoms here, we want this to be all saturated hydrocarbons. So the net is we're going to convert this carbonyl into a methylene group. And we're going to do that with our reduction, dehydration, reduction chemistry that we're going to see in our upstairs level of fatty acid synthase. So I'll probably let that play because we haven't talked specifically about those enzymes, but what you'll see, watch these red carbon atoms go from a carbonyl here to an alcohol to an alkene, and then an alkene to an alkane.
The acyl carrier protein delivers the chain to the modification compartment where beta-ketoacyl reductase reduces the carbonyl group by adding electrons donated by NADPH. This results in a hydroxyl and a hydrogen now attached to the carbon. Acyl carrier protein then delivers the chain to the dehydratase, which removes a water molecule and creates a double bond between the two carbons. Acyl carrier protein next delivers the chain to enoyl reductase, which uses NADPH to reduce the double bond to a single bond. The growing modified 4-carbon chain is then transferred to beta-ketoacyl synthase, and the entire cycle, beginning with the attachment.
So I'm going to rewind for a second here after I say a couple things so you can see this. So acyl carrier protein then kind of went upstairs and did all of its modifications, and then that group is ready to be an electrophile again. So that's going to get transferred back to that Ks domain, ready for more condensation. Then that acyl carrier protein is going to pick up another malonyl-CoA, which is going to be a nucleophile.
Is then transferred to beta-ketoacyl synthase, and the entire cycle, beginning with the attachment of malonyl-CoA to the acyl carrier protein, is repeated until the fatty acid chain.
And they were just kind of showing we're repeating this. So this is going to be repeated until we get to be 16 carbon atoms long. So I'm going to rewind this for a second.
CoA to the acyl carrier protein is repeated until the fatty acid chain contains 16 carbons. Thioesterase cleaves and releases the 16-carbon fatty acid, also known as palmitic acid, from the acyl carrier protein. In this way, multiple enzymes come together to create a long-chain 16-carbon saturated fatty acid from molecules of acetyl-CoA and malonyl-CoA.
All right, so I hope that helped kind of put that previous sort of static chart, if you will, which were showing the different domains of fatty acid synthase into a little bit more of an applicable sort of way. So that's chemistry of fatty acid synthase.